Distinguished faculty members and dear participants, a very good morning to all and all. I cordially welcome you all to the monthly faculty development program on exploring the potential of natural resources in Pradesh development. Now, let us welcome our guests on this. With the great privilege and honor, I request our principal, Dr. S. Vidyadhara sir, to come on to the dais. Due respect, I invite the conveners of today's session, Dr. J. Chebrolu sir and Dr. V. Venkataraw sir, on the dais. Now, it's a great honor for me to invite our today's guest and speaker, Professor Y. Rajendra Prasad sir, on the dais. Now, I invite Dr. V. Sindhuvaishnavi to come on to dais for a prayer song.
Manasa smarami Mahaganapatim Manasa smarami Vasishtavamadevadimanditamaha Ganapatim Mahadev Mahakavyanatakaripriyam Mushikavahanamodikapriyam Mahakavyanatakaripriyam Mushikavahanamodikapriyam Manasa smarami Vasishtavamadevadimanditamaha Ganapatim Manasa
Now, I request our principal sir to give a welcome address for this particular meeting.
Very good morning to all. Today is most auspicious occasion that the most learned personality, our well-wisher, popularly known as Professor Y. Sir, has visited our college after almost a gap of 2 months. Have you ever seen Professor Y. Sir at our campus when he joined reform during 2020? You have not seen. I am privileged to mention that I am direct student of Professor Y. Sir. when I was pursuing my B.Pharmacy at Real College of Pharmacy. So under his able teaching guidance, we were all groomed to this level. And at the same time, he is always very happy to mention that since its inception of our college, Recruitment of Pharmaceutical Sciences in the year 2005, there was an acute shortage of faculty members for teaching, particularly the pharmaceutical chemistry subjects. So when I requested Professor Vayati Sir for the first two batches, he immediately accepted our request. He used to come almost every month once to our college. He used to teach all chemistry related subjects to your very first two batches of students. That led to the grand success of the first two batches of students with 10% distinctions. That was the greatness of Dr. Vayati Sir, who always been with us, mentored our institution, guided all of us in all the parts of our institution development. It is not only guiding us. There are your faculty members who are sitting on the dais as HODs, Dr. Venkatrao, Dr. Subbarao, even Dr. Chakravarti. All these are encouraged and got PhDs just because of Dr. Vayati. And I am very bold and excited to say that I am being working as BOS chairman at Andhra University for the past 9 years. It is all just because of the blessings of Dr. Vayati Sir. This is the courtesy that Vayati Sir always shown on our college, and he always supports our institution in all walks of our college growth. And he always likes to interact with the students rather than giving speeches on the dais or on the platform in a public place. So he is always comfortable if he is given a blackboard and chalk piece without any paper. Very fortunately for this batch, there is no chemistry of natural products. So when we were studying, when your seniors were studying, chemistry of natural products are there. In that there are so many complex structures and complex mechanisms and reactions are there. All these. Still, I am very well aware. Just because of Vayati Sir, I could able to learn the synthesis of cholesterol, which is having 17 steps in that particular reaction. He used to write that synthesis of cholesterol without any paper on the board. That is the caliber and capability of Vayati Sir. And not only that, if I am right, sir, you have completed successfully and guided and mentored more than 95 PhDs so far. Maybe in next year or by 1.5 years, it's going to achieve a landmark of 100 PhDs under his able guidance, and we are going to witness that particular also. So what I am saying is, so we are always should learn the things from a learned personalities like Vayati Sir. So he always give very simple and easy tips to the students how to do the research and how to execute that particular research. So initially, from our college, any proposal reaches to Dr. Y.R. sir, without any hesitation, he used to say, go ahead, I am there, I am going to help you, and you will succeed in that. In the same way, we are successful under the able mentorship of Dr. Y.R. sir. And for the last couple of years, just because of pandemic and just because of his busy schedules, now he is serving as principal of the most prestigious Andhra University College of Pharmaceutical Sciences for the past four years. Andhra University College of Pharmaceutical Sciences. And at the same time, he is always busy and visited Australia and America to see the welfare of his daughters. And just because of his all busy schedules, we could not able to invite him physically to our institution for the last 4 to 5 years. And it is very fortunate that today he accepted to visit our college. He visited our college and he wanted to interact with you all on this August occasion that we are going to conduct 1 week hybrid mode faculty development program. So you may be getting doubt. The title is faculty development program and you are all forced to sit here. It's not that. It's not only for faculty. Since you being the outgoing students and your aspirations on several facets of research is very, very important. So for that particular purpose, now most of the research is mainly focused on natural resources. So for that, our pharmaceutical chemistry and pharmacology departments have come forward to conduct faculty development program on the title Exploring the Potential of Natural Resources in Drug Discovery. SAR is expert in both parts of research, that is synthetic chemistry as well as natural chemistry. So in Vizag, particularly in Andhra University, most of the professors used to do the research, particularly on marine resources. SAR has conducted several marine research activities, got patents, and at the same time is equally capable of doing research on synthetic products also. So, since SAR is having extraordinary knowledge on all these aspects, it is appropriate to invite and inaugurate this particular 1 week FDP program under the blessings of Professor Y.R. So that's why we invited Dr. Y.R. and also to bless you all since you are going to go out for your future endeavors. So getting blessings from such a dynamic personality is always beautiful. You are all witnessing here. So many people are here. They are all blessed by Dr. Y.R. and they are all now prosperous, including me. So definitely with these blessings, you are all going to be prosperous in your future endeavors. And this particular FDP program enlightens several research potentialities on natural resources from various experts, starting from our parents. On this August occasion, I once again cordially invite and welcome Dr. Y.R. sir for immediately accepting our invitation and being with us today for addressing you all. Since it is online program, there are more than 200 participants are visualizing this particular program, and this particular program is live streamed. Okay, more than 250 participants are already along with you are visualizing this particular program. I once again welcome all the online participants to be a part of this particular FDP, and make use of this facility and learn the expert talks and utilize this opportunity. I welcome all the online participants as well as offline participants, particularly our outgoing students as well as our faculty members and everyone to make this particular program a grand success. So with these few remarks, I once again thank Dr. Mayappi sir for immediately accepting our invitation and being with us to address you all on this FDP program. So you know pretty well that our institution is regularly conducting several programs for the benefit of students. They are all maybe workshops, maybe seminars, maybe guest lectures. And faculty development programs we have conducted during the lockdown period. We have conducted several faculty development programs online. Why? Because we have got rich resources to conduct faculty development programs through online mode since we are having June subscribed platform. So through that we have conducted several online faculty development programs. So this year we started faculty development program with the Association of Chemistry, Pharmaceutical Chemistry and Pharmacy departments, and they are conducting this particular one. I congratulate both the departments for choosing this apt topic for the welfare of all the pharma fraternity who are involved very much in research activities. So with these few remarks, I once again wish you all to stay back and listen carefully and at the same time gain knowledge on the expert talks that they are going to deliver. Thank you, Andhra.
Now I request our today's guest and speaker, Sir Mayappi sir, to please come on to address our students.
Chowdavaram, as he rightly mentioned, my student at the academic Brad principal at Chebrolu. My other students on the dais, Professor Venkatrao, Professor Sobrao, Madam, Lieutenant Faculty, and students. Very good morning to you all. It's a moment of happy welcoming me to this College of Pharmaceutical Sciences. As Dr. Chowdavaram was mentioning, I used to come very frequently in its initial years of formation, at least once in a month. I used to address the students. Particularly, I like teaching first year students because what I observed over the years of my 35 years of experience is the students nowadays lacking basics, particularly in subjects like pharmaceutical. I also observed many students not knowing what is pharmaceutical chemistry, what is pharmaceutical and what is pharmaceutical chemistry. Because pharmaceutical is called to almost all the subjects throughout his career in pharmacy. So I need to highlight the difference between a drug, between a pharmaceutical, between a bulk chemical, between a fine chemical. I very much remember having been taught, which used to give satisfaction to me, but also the students. because they are the building subjects like physiology, anatomy, pharmaceutical chemistry, physical pharmacy, all these subjects, unless you are thorough in those basics, you cannot read with a clear understanding or digest with a clear understanding the subjects that follow that. Second year, third year, or final year. So that is very important. I was fortunate therefore to come here and deliver such lectures. And also, as your principal was rightly mentioned, this is a college where you find number of seminars conducted. In fact, he used to invite me whenever a seminar is being organized. But for the last 4 or 5 years, as I was totally busy with the administration there, I could not find it to come here, even though he invited me. So I thank the principal for inviting me to this important seminar on exploration of exploring the potential of natural resources in this. I'm always interested to speak on natural resources as source of drugs. So once they invited me, I felt happy to share some of my experiences on natural resources. India, particularly the source of many medicinal plants from different geographical regions, still untapped for their potential as to be used as a potential source of drugs. Some of the plants have been exploited, but if you look at particularly in Andhra Pradesh, you have eastern Ghats, where a variety of flora and fauna you can find. 80% of this flora and fauna yet to be touched, which speaks of the potential for the drug discovery. When no much knowledge was accumulated on drugs from synthetic sources centuries back, people needed to depend on only plant sources. They do not even know that this plant is having any medicinal effect. It is by trial and error because they need something to recover from the disease from which they are suffering. So they used to take a leaf, a root, a flower, or a pod, or different parts of the plant with a hope to get the benefit by blind methods. Or sometimes based on the observations of the tribal people who used to take these plants and used to get the benefit. To some extent, if not to a large extent, provided the required benefits. And then they thought when a particular plant is providing the required benefit, why not we refine the way in which we use the plants? Plants are also toxic if you do not carefully establish the safety of them. So then the next step has come to extract the plant principles by a suitable method, say with water, alcohol, or some other solvents. So that started. People in fact used to take in those days what is called decoctions. And then started extracting the active principles. You know how the extraction of active foods has led to many natural products which you are studying in your people. You know, the atropine from metropa a receptor from raw material serpentine, with crystal with blast from rosea. The number of such drugs you can name to any extent. They have contributed to the drug discovery program. And the next advancement came, even though they have some activity when you take inside. The problem is of different types. They may not be having the required potential, required level of potential. The question is how to increase the potential. The second problem is they are not able to reach to the place of action where they should go. How to make them accessible at the site of action. The third one is the toxicity which they display. How to reduce the toxicity. So when they are not having the required level of activity, what people usually do was to change them into a suitable form by a simple modification. We usually call them as semi-synthesis, which could provide big leads. You can see the local anesthetic drugs today we use, like procaine, benzocaine, lidocaine, whatnot. The original drug was cocaine from the leaves of Erythrogenium coca, which used to be very active as local anesthetic, used to be applied in ophthalmic preparations, but possessive toxicity, corneal toxicity. So later was modified and the proofs they found are able to prepare drugs like benzocaine, procaine, all these based on the structure of cocaine. Had cocaine not been isolated, we would not have got all these drugs, isn't it? So this is a contribution of semi-synthesis, not only with respect to cocaine. You see, for example, morphine. Are you using morphine for the years now? No. The reason being its addiction and tolerance, misusing the drug. So based on the structure of morphine, we are able to get a number of analogs of morphine, which are not having the side effects of morphine. Like this, the antibiotics from microorganisms. The original antibiotic was penicillin, penicillin. Even though it is being used as a life-saving drug in some cases like pneumonia, still lot of disadvantages are there. Drug resistance is there. Also with all antibiotics. So the need is to convert them into more and more important antibiotics by some semi-synthetic methods. Because nowadays semi-synthetic penicillins, a range of semi-synthetic penicillins, starting from amoxicillin, ampicillin, cloxacillin, dicloxacillin, whatnot, and other antibiotics replacing them because of the increasing resistance, misuse of antibiotics. So all these are developments necessitating the potential use of natural resources as per the requirements. Still there is a lot of scope for natural resources because even in Western countries like Germany, UK, even in the USA, lot of research happening on natural resources, particularly as potential anti-cancer agents. Even if we use many synthetic drugs to treat cancer, you know the problem. The toxicity associated with it, no selectivity, and no complete relief whenever a cancer occurs, unless it is realized in the early stages. You cannot have the complete cure. Similarly, for other diseases, disorders like diabetes. What we are able to do, we are only able to alleviate the symptoms of the disease, but not the underlying causes. Rheumatoid arthritis, very inflammatory disorders. We are only reducing the intensity of the symptoms, but not the complete cure. So it speaks of the fact that an intensive, systematic, focused efforts on the exploitation of natural resources need to be done, particularly in countries like India, where a large percentage of poor people not able to afford the synthetic drugs, or many people not able to tolerate the side effects of the synthetic drugs. So, if we carry out systematic research, there is always a possibility to discover and develop newer and newer molecules with less side effects, because these molecules are biosynthesized in the plant sources. They are not expected to be so toxic like synthetic drugs. They are biocompatible. And hence, research is going on to a great extent on natural resources, of late on marine natural resources. Because marine organisms under the deep sea, they are able to survive against high environmental pressures of the sea, against invading pathogens, against the salinity of the sea. The interest is how they are able to survive. There must be some metabolites in the system which are able to protect these marine organisms growing under the deep sea. So they are having drug-like properties. Why not we collect these marine organisms under the deep sea and extract those principles and try to use them as drugs? That in fact has provided greater leads for some of the diseases which we are not able to address from other natural sources. For example, cancer. Many effective drugs have come from very natural sources. So like that, a number of different natural sources are being examined and potential. And it is in this context where we are addressing the problem, exploring the potential of natural resources in drug discovery program. I congratulate and thank the organizers for identifying their theme, and I wish you all the best of luck. And I thank the organizers for inviting me as a resource person to this work. Thank you. Thank you.
Now I request Dr. J. Chebrolu sir announce the 1 week program schedule for all the participants. Thank you, sir. Thank you, madam. Respected dignitaries, organizers, and organizers, my dear students. In a hybrid mode from 18th onwards, that is today onwards, to 23rd, we are conducting combining that is pharmaceutical chemistry and pharmacology department. 1 week faculty program, faculty development program. That is today, 18th March, natural approaches in pharmaceuticals. Tomorrow, 19th, a review on phytosomes as an emerging nanotechnology platform for topical delivery of bioactive phytochemicals. In 20th, recent approaches in natural products, research for drug discovery. And 21st, phytosomes, a promising approach for the delivery of phytoconstituents. and 22nd, drug discovery in biopharma, and 23rd, drug discovery from natural sources, that is approaches and applications of various natural products. So this is the schedule for the faculty development. Not only the faculty, for the outgoing students also it is very useful. That's why we were also involved with this program. Thank you. Now I request all the dignitaries on desk to felicitate our chief guest. Now we go to the today's session. So before starting the session, now I request Dr. V. Venkata sir to introduce our speaker.
Very good morning to all of you. Once again, on behalf of Chebrolu Institute of Pharmaceutical Sciences, I welcome you all to day one of the faculty development program on exploring the potential of natural resources in drug discovery. It's a great honor for me to introduce today's resource person, Professor Y. Rajendra Prasad. Sir completed his B.Pharmacy in 1984, M.Pharm in 1986 in pharmaceutical chemistry specialization, and PhD in 1992. The entire education is from Andhra University. So during SAR's post-graduation program, SAR received university second rank. SAR is currently working as principal AU College of Pharmaceutical Sciences, Andhra University. SAR is having total 34 years of teaching, research, and R&D experience in the field of pharmaceutical sciences. Previously worked as a research officer at Speak Pharma in Herbal Drug Division near Chennai, and worked as UGC Research Fellow at Andhra University, Visakhapatnam for 3 years. And SAR research areas of interest are chemical studies on bioactive metabolites from natural sources, synthesis and evaluation of bioactive heterocyclic compounds, development of synthetic routes for modifications of bioactive natural products, development of analytical profiles for drugs by using HPLC and RPHPLC and other new hyphenated techniques. During his 34-year long career, SAR
you, Professor Abraham, the next person. Actually, visionary works are just the beginning at Andhra University, like Dr. CRD, Sarvepalli Radhakrishnan, and Dr. B.S. Krishna. Why? Because they used to invite eminent faculty from not just from Andhra Pradesh, from across the country. Like that, stalwarts came to Andhra University in different disciplines offered at Andhra University. That has led to the joining of great people like Professor P.R. Seshadri. No longer a replacement happened so far in natural products. He headed the Department of Chemistry at Andhra University, the only FRS chemistry so far. Then Professor S. Rangaswamy is a student who headed the Department of Pharmacy. These two people, they have created a very strong environment for natural products research, which sustained over the years and even up now. Many people follow, like L.R. Rao, like Professor Devasankar, Professor Anjali, my mentor, Professor E. Venkatraman, many people. In fact, in our country, if you look at the number of drugs discovered, it is very disappointing. There is, or there are not more than 2 or 3 drugs so far. One of the first drugs discovered in India is from the pharmaceutical chemistry laboratories of Andhra University. A joint drug discovery by Professor E. Venkatraman and Professor S. Rangaswamy is there. The isolation of the cardiac glycoside, peroxide, from the fat Tibitia peridea still being used in countries like Italy, like desoxyribonucleic. A monumental drug discovery from natural sources. Even though many drugs later came no longer being used or no longer a single chemical entity, like a mixture of drugs like Pharmaceutical, so on and so forth. And the other synthetic drug perhaps only one is the Chandramoulipuram by Professor Akrishnan. Punjab. That is also not being used nowadays as neuromuscular black. Except these two, the scenario is not going to agree. But a lot of focus is being placed to provide some boost to natural products. I will try to put the status of natural products research in this 45 minutes or so lecture. And if you look at the title is a natural approach to pharmaceuticals. See there, you can find different natural sources, medicinal plants, minerals, animals, or natural all these things let's see as a source of natural. What are the important natural sources for drugs? Certainly, it is the terrestrial plants contributed immensely, animals, where you get many hormonal products, even insulin for a long time, marine organisms, microbes, mineral sources, blood and blood products. We can see microbes giving rise to antibiotics, anti recombinant DNA technology nowadays we use to produce a number of medicinal substances like insulin, mineral sources, many of the inorganic compounds we use, blood and blood products, a number of antibodies, vaccines, all these we get from blood and blood products. So what is the driving force for national production research? If you look at, still there is considerable market share. 25% of the market share is from natural sources, which is a driving force to carry on research further in this area. Unmet therapeutic needs. Even if we are using so many drugs, are you able to cure all these diseases fully to satisfaction? No. That makes it important to address the untapped therapeutic needs. Still, you have to go for better drugs. For potential sources, natural sources. Diversity of chemical sources. You do not know what is really present in natural sources unless you isolate and prove. So the type of chemical structures, they are very diverse. Provides tools for people to synthesize molecules based on diverse chemical structures. Favorable drug-like properties like favorable molecular weight, small molecules. We get the required solubility characteristics, log P, that is the hydrophobicity or incompatibility, hydrogen bond, donor and accept bonds, which are able to attract exit bonds, the required number found to be there, low number of rotatable bonds, so that they will not change their configurations. High biochemical specificity, that they are synthesized by living organisms in response to selective pressure and represent a population of related structures. Because they are biosynthesized, already possess some drug-like properties in the plants or microorganisms. Expected to have drug-like characteristics. Diversity of biological activities. We do not know what type of activity. In fact, if you see the natural sources, different molecules, they are being used for a different types of activities. Development of novel, sensitive, and effective techniques of isolation. 2 decades ago, the technology was not handy to isolate even the minor molecules because there was no chromatography techniques. We do not have HPLC and some other methods or spectrophotometric methods like NMR, which we are having today. So the advantage, we are at an advantage. So development of novel and effective techniques of regulation, similarly purification methods and methods, and the characterization of belongs to better methods. But since some technologies in the global being increasingly used in the Western countries. So all this being very close to natural resource research. As our type say, exploration of potential of natural. This is the driving. At the same time, we look at the upside of the coin. What are the things in value for selection of plant? We do not know exactly how to select the required plant. What is the basis for the selection of this plant? Proper authentication. Even if you select a plant material, how to authenticate whether it is the same plant which we intend to collect, because number of species variation in same genus, we find. For example, Phalanx, there are number of species, even though Phalanx, very important. There are number of species which comes along with them. They are not useful. Limited supply. You cannot get the required quantity. Threat to biodiversity. If you want to collect in large quantities, you have to sacrifice the plant. The threat to biodiversity. Seasonal variations of preference. At which season we have to collect the plant material. Place of collection. What is the right time, right place need to be established. Correlating the biological activity of the original extract fraction to the feed form. Your extract may be active, your fraction may be active, but once you isolate a compound and try to correlate the activity with its plant, several cases we miss them. What is lacking? Why the substrate is not added? Why the extract or the fraction is added? Means there is something else which is required. Maybe two or both substrates need to be combined, or something else which we could not isolate may be present in the extract or fraction that may be responsible. All these make up activities in establishing or correlating the activity which we are looking at for the risk. Laborious, cumbersome, unproductive, and time-consuming. We usually to spend a lot of hours, number of hours in the laboratory, training the plant material, doing extraction, using large quantity of solvents, wasting a lot of time. But when you see the result, it is not worthy, not useful, unproductive. We use it to throw them in. The chance of getting a compound, useful compound, very, very limited. The expenditure involved is not less because you are wasting many resources without any benefit. So these are some of the hurdles in natural resources which we need to address effectively. So what are the subjects to be integrated for productive research in natural products? To carry out effectively the research in natural products. What type of understanding as pharmacists you should have to carry out this research very smoothly, very effectively? Pharmacology. You already know that pharmacology is the subject which deals with the natural resources, natural plants, covering various aspects: cultivation, collection, identification, histology, identifying the constituents, hereditary, all these aspects you do study in pharmacology. And then natural products chemistry. Once you isolate the component, how to establish the structure, to have separate subject like chemistry of natural, wherein we study the methods of isolation, the methods of identification, the structural elucidation, either by spectral or by degradative studies, all these we do study. Medicinal and analytical chemistry. How to use them as drugs, how to estimate them by a proper method, because when you use them as pharmaceuticals, you should have also methods to estimate these drugs, how much is present, or to analyze the purity. Biochemistry, to understand how these molecules interact. Pharmacology, we establish their activities by different methods of the preliminary blind screening. Pharmaceutics, you need to develop them into formulations by proper methods. So all these subjects need to be understood fully. And then I told you natural sources, they provided like drugs, overweight. If you look at the contribution of this drug discovery from natural sources, we cannot just from one source. They contributed to the drug discovery and development in different ways. One is by direct extraction of the plant. You take the plant, extract with the solvent, and then isolate the compound. You need not do any modification. We establish the structure. That way, we could get drugs like many cardiac glycosides, redoxin, ketoxin, mercurium, alkalies, I need not mention the number of alkalies, metropin, the metropol, belladonna, the second, the problem, wind crystal, wind blast, the number of alkalies, cataract, metropin, morphine, all these things we could get simply isolating and using the methods. We need not carry out any more. And then by acting as a prototype structure or as a read, means we get the molecule, we have it reacted for some problem. We need to overcome that. For example, based on the structure of cocaine, the number of local anesthesia. Morphine, simplified analogs are developed with better pharmaceutical and therapeutic properties and with less side. Pharmaceutical means they may not be having required soil. You have to enhance the soil. You have to put in the techniques like conversion into salts, conversion into esters. They are pharmaceutical therapeutic properties, increasing the active by making structural molecules with less side effects. So that way we have got a told you already from cocaine to vegetable, cocaine, diclofenac, because cocaine is a very complex molecule obtained from the leaves of the retrograde, very complex structure. That to having some toxin. You have to sacrifice the plants. So what they have done is they have identified the structure. Is it really required to proper structure? Or only a part of the structure is required for it? Only a part of the structure is required. Why not we use that part instead of the proper structure? We call that as essential structural unit. Essential structural unit. So what they identified in cocaine is, a nitrogen is required, an ester group is required, an ester linkage between two molecules is required. So then they prepare drugs like benzocaine, having a benzene, having a ester molecule, COOCH5, and then nitrogen in the form of NiN. Photolysed synthesis. How good it is? It really works. So similarly, what the number of analogs. So like that, this is another dimension how the natural products start. By demonstrating a mode of action which is their carbon. For example, discovery of neuromuscular glandular agents like Chebrolu from arrow poisons. People used to hunt animals by using arrow poisons. The animal will fall. What is the problem? They thought narrow budget reactions of blanket. So they identified the active principles and used them as neuromuscular blanket. Had you not found that neuromuscular blanket property, you would not know about it. So that is another way to indicate the discovery of drugs plants. Similarly, by providing a starting material for the synthesis of a number of naturally occurring hormones. For example, a number of molecules which we isolate may not be active at all, but they are available in large quantity. If we make a small modification on this molecule, that becomes settled. For example, if you see diastereomere, which we obtain from Diastereomere species, a steroidal molecule, a saponin. It is having a structure very close to progesterone, the female sex. Today we get the progesterone not by synthesis. We get from diastereomere. To take diastereomere, make a simple modification to convert it to progesterone. And then number of specific penicillins with broad spectrum of activity and with less resistance by taking a starting molecule 6 amino penicillin which we obtain by downstream fermentation technology from the molecules. We convert that 6 APA into whatever type of semi-synthetic penicillin we require. A number of peptide hormones like the insulin today owe their origin from animal species. But nowadays we are getting by recombinant DNA technology. The unusual antiviral drug RA and anti-cancer drug RC are obtained from the unusual nucleosides fongoiodine and strombothymiidine isolated from the marine sponge Tetia. Another dimension, the marine sources. Not just these molecules, operate the number of bioactive molecules from marine sources increasingly being isolated, being used. What are the factors to be considered for drug design based on lead optimization? I told you sometimes we get a drug which is not having totally the required potential. You need to optimize its activity. If optimize its solubility, reduce its toxicity. How to do it? We want to improve the binding interaction. When you get a toxicity, what is required is any drug for that matter, even a synthetic molecule. Then we take how it produces its activity, the fundamental properties, which should attract the receptor which are nothing but proteins. So you have to enhance the interaction. to increase the activity. So we improve the binding interactions between the drug and its target in order to enhance it. But it is not just substrate interaction. To increase the drug selectivity for the required target. When it is interacting with other targets which are not required, we get side effects. So what is important is the required selectivity to the required target. That we have to take into account in our sheets. To design drugs that are as easy to synthesize as possible. It is of no use to synthesize to design a molecule which is very complex and say this is avid active. If it is impossible to synthesize, what is the use? So to design drugs that are as easy to synthesize as possible. Otherwise, it is useless to design drugs that are impossible to synthesize. Another one is to design drugs with optimal pharmacokinetic properties. That is, whatever drug we design, when you synthesize that, it should reach the site of action, which is what is required for that proper absorption, distribution, metabolism, and excretion plan. So if you do not have that, only you have in vitro favorable properties. When you administer individual, it pays to you. So that is also very important. And when you take a medicinal plant, how should be converted into a drug? You see, it's not an easy process. It requires a series of steps. You have to identify the right medicinal plant, which include classification, we do repeatability. Then we collect the plant material, separation of individual reports, and then we write them based on the properties of the plant. Where some volatile constituents are there. We cannot subject it to sunlight, sometimes we do artificial rain, or shade rain, and then we powder them. Because when we powder the plant material, the extraction becomes very easy because the surface area becomes small. So we powder them, we extract again by different methods. One is hot method and the other one is cold method. Where there are some thermodynamic substances, we cannot do hot extraction. We use cold extraction by taking them to the laboratory. And then once we extract them, we go for separation of the constituents from the extracts and then unify them by observing them on the PLC, electrophoresis, GLC, even carbon chromatography, all these techniques we use to get a pure compound. Once we get a compound, we identify if it is a low compound, we compare it with a reference compound. If it is alcohol, we establish structure by different methods like identifying its melting point, spectrum, IR, UV, NMR, mass, elemental analysis, elemental addiction, PLC, we establish the compound, identify the compound. Then go for biological activity. It includes pharmacology or microbiology. And then if it is promising, we go for clinical trials, phase 1, phase 2, so on and so forth. We establish the clinical utility and then put it in the market, put it in the large scale survey, and then only we get the required market. In between, it is failures. So it's a very difficult process to get it right, even from a synthesis also. Costs for an expenditure of $1 billion on an average. So what is the basis for selection of granted? You see, how do you select? The information from material medical of traditional remedies in various cultures like the writings of diasporites in the first century AD, you call it the pre-material medical. Writings of Germany, the second century, Chinese material medical, or Ayurveda, information from local tribes, or simply by blind search without any basis. How literature opinion is useful in graduate research? Provides the scope and guides the chemist to go or not to. When lot of work has already been done on structures established to the fully local level and out no use. There is no use to go in. Where's the structural investigation is? Because you already got some information on the chemistry of the molecules by literature review. When you isolate some more molecules, they may be closely related to the one which is before, because they are all biochemically synthesized. So to establish the structure, it is very easy just to compare with the known molecules, how this structure is different. Without having that knowledge, you cannot go into the blank and establish the structure. So that is another use. Provides insights into the possible biology. Because you also know from that literature what type of biological activity and what these molecules are expected to have, which may be better than the explored biological activities. Specific methods, modification of methods of isolation will be followed in view of the type of molecules. If your interest is to isolate the molecules which are present in very, very small quantities, as based on yields, minor quantities, you should modify your methods of isolation using a different type of solvent, which can extract the. So that also we get from literature. Increases the interest of the chemist to look for some more biologically and chemically real minor sectoral. If necessary, by using large quantity of and by using most decent methods of substitution. This in fact has given more and more molecules from the plant. The question is whether dried or fresh plant material. What is the choice of dried? So if a thermolabile or thermostable constants are there, we cannot certainly go for sun drying or drying which the plant material is exposed to high temperatures. So therefore we have to go for a other type of. We cannot dry them, so we use the fresh plant material. Volatile ion structure. Here you cannot go for a dry. Polar, non-polar constants, one is interested in. If you are interested in polar constants, we go for one type of dry. We go for directly fresh plant. Non-polar means the plant material contains a lot of tissue water. Unless and otherwise you dry the plant material, when you extract with a non-polar solvent, determine whether or the other. That cannot penetrate into the tissue. So you have to dry it, then you have to extract. So that those things you have to take into account. So what are the methods of extraction? See. Stepwise extraction and direct extraction. If you see stepwise extraction to follow when you dry the plant, where there is no moisture present. we extract first from Chebrolu. So most of the non-polar functions come here. And then if some more are there, we use Europe. And then we go for Hanumaiah, which direct extraction we use by directly taking the fresh plant material, which contains a lot of water. We extract the charcoal because the water present in the plant material is also visible out, no problem. So we use the direct extraction method to dry the plant material, but by the fresh plant material. That we use. Shampoo extraction and direct extraction, they are the only two methods we use the methods of under the methods of. Once we extract, then what to do? We do the qualitative question. We do the study in part of this year, chemistry of natural. Qualitative question. What are the type of compounds? Alkaline, flavonine, carbohydrates, proteins, steroids, terpenes. There are specific tests by using specific reagents. So we go for these tests to establish what type of compound. How many compounds are there? What is the complexity? We simply place a spot on the PLC, relay protocol, and run the plate. Relay protocol PLC chopper, and then spray the plate with a spray reagent. We get number of spots. How many are there? What type of colors we get? It speaks of the type of compounds we get also. Vapor chromatography, parallel to PLC, we use vapor chromatography, acidic or acidic vapor chromatography. HPLC, very recent method, which is a nowadays it's simply fingerprinting the plant extract. Hydrogen three-layer process. And HPS, hydrogen liquid process, in the pharmaceutical. Very useful to isolate those compounds which are very closely related, which could not be isolated by other methods. We use. Fractionation of extract. How do you do that? What are the procedures to be followed? When you extract, the problem is the petroleum extract, the chloroform extract, alcohol. They contain 100 compounds per cell. Is it possible to directly isolate from this extract? No. When you have one or two compounds, we use to separate them by crystallization, fractional precipitation, so on and so forth, by distillation. But when the extract is very complex in nature, we try to reduce the complexity of the extract by what is called fraction. That is, we convert this extract by removing the solvent and then adding slowly water. We take it to an aqueous state and take this into a separating panel. And to this, we use organic solvent because they are mixed. We shake them so that some of the constituents which are non-polar will come into the either or chloroform and two layers of them. We touch the organic layer. We repeat that process until the organic layer is not having its color. We combine the organic layers, we distill out the solid, we get the organic layer. That will not contain all the components which are present in the original. The complexity is very much better. Like that, we take the again the original one, the mother vector we call. We again take it with the separated project, shake the solvent, chloroform. Like that we do, we get different reactions. The advantage being the complexity is reduced, the separation process becomes easier. That is very much important in natural process. Extraction and fractions. So the procedures I already told. And then once you get a fraction, again to identify the components, along with the original mother method. Original extract and the fraction is possible in TLC or Java. We can easily find the difference. The all molecules present in the original extract. What are the less number of components present in the TLC in that particular fraction? It guides you. And then we subject them to column formula. Here we pack the column, which is like a period, large size period. We pack them with silica gel and then add our plant material on the top. We run mechanical elution. We elute the column with different solvents. We collect the fractions. We disclose the solvent. We identify the components from time to time by PLC. When you get a single compound, you crystallize it. We keep it for identification. If it is a mixture, again we run the column. We separate them till we get a single compound. Different methods can be used. If it is not possible, we cannot consider it. If they are very closely related, we go for HPLC for using different preparative columns. Preparative PLC, where we use the PLC to separate the components by applying the substance as a bag. We run it, that divides into two bags. We cut the bag, extract with the solvent, and then filter that as dryly solvent, remove the solvent. So that is preparative PLC, HPLC, by a similar method. Quantitative HPLC we call it, and then finally make it 100%. So very difficult job to get a clean compound seven. It requires a lot of skill and efforts. Natural products are structured determination. You see, the purity of the compound is established perfectly pure as evidenced in the TLC and melting point. Sharp melting point. Then we go for the UV, IR, NMR, mass spectrometry. It gives you the spectral character. You know the use of each and every method. UV gives you the type of saturation of the silicon. IR gives you the functional like hydroxide, whatever. Apart from all these groups, NMR is the hydrogen in environment, which carbon is attached. What are the carbons, type of carbon, whether and then mass spectrum, what is the important molecule of each of them? Whatever structure is used, we map into that molecule name. So all this is important. Running a 4 PLC with authentic compound will be the wrong. For example, if we see the molecule isolated, we got the proton signals to identify melting point, neutron, IR bonds. We got it. Right. Then isocarbon. Then it's mass, how it is degraded into fragments. It is expected to have this type of fragments with the structure is planned. We got this fragment IRs in the mass spectrum. You have to identify. So if it is correct, this structure is correct. Natural products and study of biological. We follow line screening of good compounds. Other studies, future studies are identified. The emerging activity out of the line studies. We concentrate on that. Future studies are identified. Screening of fractions and identification of bioactive. When you screen number of fractions, which fraction is having them? We consider that as bioactive. We need not isolate, go for other fractions. Only bioactive. Isolation of compounds from bioactive. Study of biological activity of the isolated compounds from these because they are bioactive. These compounds are expected to function. And then we go for preclinical toxicity evidence. They should not be toxic. You have to establish already. And then formulation. And then clinical evaluation in phase 1, phase 2, phase 3, and phase 3. So, what is the current status of plant-derived substances? The summary. About 90% species use real drugs. What political compounds of which 90 were discovered by laboratory studies conducted on medicinal plants using traditional medicine, involving a isolation, separation, and heating. The plants containing partially purified secondary active instincts were used. It is not just single molecules, but enriched fractions are also being used. Factors involved for improvement of plant drugs other than pure. What are the data? What is the data you have to cover? Parts to be used in processing. Which part you are using, what type of processing you are done. Analytical procedure to estimate, to establish the structure and activity. Indication, dosage, mode of administration, side effects and drug interaction, efficacy. All these you have to follow. Of the 32 best selling herbal extracts, only 6 are present. So if you see in this table, the natural products discovered are developed in India. Perovskite evolved pharmaceutical chemistry and other things about it. 58 in fact because CSIR created in India, which is a cardiac product being used in Italy by manufacturing sites. AMIC from Hindustan Antibiotics Limited, antifungal antibiotic no longer being used. Corticosteroid, the anti-hypertensive and cardiovascular which was used for some time but no longer nowadays used. Salai Google by R.R. Jammu, that is Pharmaceutical. And then anti-inflammatory Pupulipid. This is Boswellia serrata, hypolipidemic. This is that is Boswellia serrata. This is other one. Dilax C, developed by CDRI, cervical dilator for medical termination of pregnancy. Putkin, by R.R. Jammu from Piprolide of Goa, Piprolide, same CDRI, all these are mixtures for additive fractions, which is used as a anti-inflammatory. Memory plus from Ecopa binary, we have mecopocides, a mixture of mecopocides, memory enhancer, produced marketed by Reward, Artemida, Artificial Eduva, which is used as anti-malarial drug. Urea Cebrolu, no longer being used. Avaja, Methaqualo is bad, hypnotic, Chowdavaram holiday, neuromuscular bracket, Punjab University, Promaril, Hyderabad, anti-inflammatory, Centirizone, anti-thyroid, all these are not being used. But some of the drugs developed, discovered in these are synthetic drugs also. Memory enhancing plant drugs, some commercial preparations available in India. Memory plus capsules, memory plus Ecopa binary extract, Chowdavaram active limited, Eca capsules, Brahmi leaf powder, Jagat Pharma, Antat tablets contains Brahmi, Madhukapatri, Vishnukrant, Jetmarsi extracts and powders by Hanumaiah. Brain tablets, Ravi, tablets for Ravi, Chowdavaram, 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 Pharmaceutical, Pharmaceutical by them. Also changes Hanumaiah, most popular contains downward electrolytes, some technique employees, increases cerebral blood flow, especially with geriatric patients. That's steady hormone medicine. Yes. Echinacea, garlic, very upside, golden seed, hydrosis, keratosis, insect, interaction, before, biloba, there are now, opposite, top organelle, allied health, hospitals, etc. Victoria, and Doctor Indian Herbal Medicine. Just standardized restaurants using just ashwagandha, very important one. Certain phenolides, herbal adaptogen, synthetic substances are Asiatica, using productive or alcohol. Treatment from Garcia, Hanumaiah, management. Contains decoction salts of hydroxychloric acid, cholesterol, anti-hypertensive, hemoglobin, cholesterol management, diminima silvestre, anti-diabetic, which contains glycemic acid, chlorine, appendix protective, shellac from mineral sources, developed by Pradesh University people. General body, very coded from levels for a particular with their directly. When you see the factors, which very little speak in the stage, gas system and gas system or oxytocin and application, but very little different types, so that's for you, no use, activity analog. They are also related to the market. Scoporin from artificial scoporin, roller reduce, propagin from propagin genica, cardiofarm, peroxide, isolated from organization to degree. The facility is not a product of the cardiac states, but also in their anti-pyrenic particularly around something Streptococcus, Lasper, Astrocyte and Stenocyte, both show significant micropyrenic activity. And the more effective molecule in the Astrocyte.
That is the last one. Last week.
Okay. Okay. So, you should know, to love Sarah and for molecules. So, still there are lot of research need to be done to address the issues which we could not do complete, like osteoporosis, arthritis, old age problem. So, need to be focused to isolate such molecules also. Okay. So, you have been exposed to different aspects of natural course research, the advantages, the limitation of this challenges for so forth. Okay. If you have any problem, that will be
Thank you, sir. This is a very useful information and the session is highly informative, starting from the original plants to the final product, which is very much informative for all the research scholars and even our final year students who are going to do their project. Most of them, 70% they are doing their work on only natural products. So this session will be very highly useful, not only for our students here and faculty, for all the participants who are viewing from online. This is very useful session for all of us. Thank you, sir. Thank you so much.
You are.